All right, great. All right, so last class we have been discussing the different parts of the digestive system. If you remember, we discussed the connections here between the liver and the duodenum, as well as the pancreas and the duodenum. A quick review of this important diagrams that we've discussed last class. If you remember, the bile gets produced from the hepatocytes, the liver cells, and through very small ducts, we're going to be conducting this bile from the hepatocytes into two main ducts. We call those ducts are my right and left hepatic ducts. The right and left hepatic ducts will then conduct the bile into the common hepatic duct. Part of the bile that we are releasing is going to keep going down, but because of the pressure created by the sphincter, most of it going to be moving up to be concentrated and stored in the gallbladder. Once I get food in here, in the small intestine, the sphincter relaxes, and this is going to be reducing the pressure within the common bile duct, and in the same time, the gallbladder will be getting a signal in the form of a hormone called cholecystokinin for it to contract, and as it contracts, it's going to be pushing this concentrated bile out to travel through the cystic duct first, and then into my common bile duct or the bile duct. The bile duct, as we've seen last class, it meets with the duct that will be conducting the pancreatic secretions. We call the duct that conducts the pancreatic secretions, this is the main pancreatic duct. Both will meet and will be releasing their secretions in the duodenum for them to be mixed with food that will be digested. If you remember, this elevated part where both the bile duct and the main pancreatic duct are going to be meeting is called my hepatopancreatic ampulla. And remember, the hepatopancreatic ampulla is surrounded by a sphincter, a muscle, that regulates the flow of the bile and the pancreatic secretions inside the duodenum. If you remember, we stated that this diagram is highly important for the upcoming lab exam. It's a 5 stars diagram. Moving on to the accessory organs related to the digestive system. The accessory organ here that we're focusing on is the liver. This is going to be the largest gland of the body. It's formed of 4 lobes. We can see in this diagram 2 of them. We've got the right lobe and the left lobe. We also have 2 other lobes that you can see from the posterior and ventral surfaces. Those are the caudate and the quadrate. So liver consists of 4 lobes. Those are right, left, caudate, and quadrate. The hepatocytes are going to be responsible to process the bloodborne nutrients. If you remember, any blood that gets drained from the stomach, from the small intestine, from the large intestine, this is going to be containing all the nutrients that you're going to be absorbing. All this blood, if you remember, going to need to be filtered first in the liver for the nutrients here, those bloodborne nutrients, to be processed by the liver cells. Another function of the liver cells will be to store the fat-soluble vitamins. And fat-soluble vitamins, those are vitamin K, vitamin E, vitamin D, and vitamin A. Those are the 4 main fat-soluble vitamins. They get stored in the liver cells. Also, the liver cell is going to be responsible for the detoxification of the toxins. So if a person, for example, has liver cirrhosis and you're giving him a medication, so you're supposed to give him 3 milligrams, let's say, of this medication. But this patient that you're treating has liver cirrhosis. So what would you do for the dose of the medication you're giving him? What do you think? Would you give him a higher dose or a lower dose? If you know already that this medication is going to be broken down in the liver, would you give him a higher dose or a lower dose of this medication? What do you think? So again, the question, a person with liver cirrhosis gets treated for bacterial infection, whatever treatments that you're giving him. But you know that this medication that you're giving him, its effective dose is 3 milligrams. Would you like me to give him a higher dose or a lower dose of this medication if I know already that this person has a liver cirrhosis? Lower dose. Lower dose. Why lower dose? Because those liver cells are not functioning as they should be. So their ability to detoxify those medications that you're given will be lowered. So the circulating amount of the medication is going to be higher. So that's why you will hear that somebody did take a medication and it caused him to develop toxicity of this medication simply because he might have a kidney malfunction, a liver malfunction. So he got the normal dose, but still he developed toxicity from this normal dose. Why? Because his ability to detoxify the toxin has been reduced as he has a liver homeostatic imbalance. Another major function here of the hepatocytes, of the liver cells, would be to produce the bile. And we produce around 1 liter of bile per day. But you do not release all this amount because the bile, if you remember, gets stored and concentrated within the gallbladder. The gallbladder is a thin-walled muscular sac located on the ventral surface of the liver. It's going to be allowing the absorption of the water and the ions from the bile for it to become concentrated. The bile will not be released as the gallbladder will not contract unless you've got food in the stomach and the small intestine. At this time, the gallbladder will start to contract, squeezing the concentrated bile to move out through the cystic duct and then flow to the bile duct as we've seen in the previous diagram. If you see on here, first the bile travels out in here in the cystic duct and then moves out to travel in your bile duct. So you get here cystic duct and then bile duct. So what is the problem if somebody did have a cholecystectomy? So a person has removed the gallbladder. What's going to be the problem here? What do you think? No reservoir, so I'm not releasing concentrated bile, right? So the bile I'm releasing, will it be effective or not? What do you think? Not as effective. So the bile gonna keep moving down from the liver cells directly in my small intestine, but it's not that effective because it's much less concentrated. It hasn't been concentrated and it's flowing all the time, not only at the time of the stretch of the stomach and the small intestine. So what those patients will develop? Person who had his gallbladder or her gallbladder removed. So what do you think is the problem? But yeah, most commonly it's gonna be a female. Most commonly it's gonna be a female who will have gallbladder stones, and because we are afraid that we've got an impacted obstruction of the bile duct, so the gallbladder gets removed. If it gets inflamed and it's accumulating bile stones. So diarrhea. So some people are saying here diarrhea. What is the problem? Diarrhea. So what causes the diarrhea? Digest rich food. Yeah, I have a problem digesting fatty food. Why? Because the bile main function is to perform something that we call emulsification of the fat. What I mean by emulsification of the fat, so if I'm dealing with a fat droplet like this, and those are my enzymes that are supposed to break down the fat. The emulsification is actually making the fat more soluble. So I will be reducing the size of those fat droplets. So what's going to happen? Here the enzymes will have much better exposure to the fat as you break the fat into smaller particles. So it's going to be easier for the enzyme to break down the fat. So what's going to happen if I remove the gallbladder and I did eat fatty meals, high fat content? What's going to happen? I would have this bloating sensation. Why? Why would I might develop diarrhea? Because if you didn't break down this fat and absorb it, who will be waiting for this fat in your large intestine? The bacteria. So the bacteria is waiting for this fat that you are not breaking down, you're not absorbing. So the bacteria will start to break down the fat that you failed to break down and would be causing gas accumulation, which results in a bloating sensation. And as the stool's gonna have a very high fat content, what's gonna happen? You're gonna have some degree of diarrhea, or what we call steatorrhea. Not only diarrhea, it's gonna be steatorrhea. A person will develop jaundice. No, we will not be developing jaundice if we've got the gallbladder removed, because I still gonna be conjugating the bilirubin into bile. I have no problem in my liver cells. My liver cells are functioning totally fine, but this bilirubin that got conjugated into bile will not be concentrated in the gallbladder as it got removed. So it will be flowing directly into my intestines. All right, so this will not cause the development of jaundice, no. All right, but if a person had an impacted gallbladder stone in here, in the bile duct, at this point, a person might develop jaundice because I can't get any more bile into the biliary tract because it's blocked by stone. So no further bile is going to be produced, and it's going to be accumulated throughout the liver. So a lot of bilirubin will fail to become conjugated into bile. So it will rise in the bloodstream, causing jaundice. But the removal of the gallbladder itself will not cause the development of jaundice. No. All right. So a person might have jaundice, and that's why he did need to get his gallbladder removed, or he did have an inflammation of the gallbladder. That's why he got his gallbladder removed, or he did have gallbladder stones. That's why he did remove his gallbladder. All right. Those reasons might cause the development of jaundice, but the removal itself does not cause the development of jaundice. You see what I mean? Does this make sense, Rachel? All right, so moving on to another major digestive gland, it's going to be the pancreas. And the pancreas, it's a combined gland. It's part endocrine gland, part exocrine gland. The endocrine part of the pancreas is it's formed by, if you remember, the islets of Langerhans. And if you remember, the islets of Langerhans, those were releasing two very important hormones that will regulate the blood sugar level. They are antagonizing one another. Those are the insulin, which is a hypoglycemic hormone. If you remember what's hypoglycemic hormone, it reduces the blood sugar levels. And the glucagon, it's the antagonist hormone that will be rising the blood sugar level. This is the endocrine part that we've discussed back in details in our endocrine system back in AP1. Today we're focusing mainly on the exocrine function of the pancreas. If you remember, an exocrine gland is a gland that will be releasing its secretions in a lumen of a tube through a duct. This is by definition an exocrine gland. And an endocrine gland, it's a ductless gland that will be releasing its secretions directly into the bloodstream. It's a ductless gland. I don't have a ducting here. This is the difference between the endocrine gland and the exocrine gland. So the exocrine function here of the pancreas is going to be carried by the SNI, and those are clusters of secretory cells that would be responsible to release the pancreatic juice that would be helpful in the digestion processes. Zymogen granules of the secretory cells going to be containing the digestive enzymes released from the pancreas. The pancreatic fluid is going to be about 1.5 liters per day. This is going to be containing water, enzymes, and bicarbonate. If you remember, one of the reasons that we've mentioned for the development of metabolic acidosis is having diarrhea. If I have persistent diarrhea, a person might develop metabolic acidosis. And if you remember, why would I develop metabolic acidosis? Because I'm losing a lot of bicarbonate. This bicarbonate is not going to be reabsorbed. What is the source of the bicarbonate that I will get rid of with the stools in case of persistent diarrhea? It's coming from the pancreas. 3 enzymes that got released in the pancreatic juice that will be responsible for the digestion of proteins. Those are the trypsinogen, chymotrypsinogen, and the procarboxypeptidase. Can you remind me of another enzyme that we've seen getting released from the stomach that is also going to be responsible for the breakdown of the proteins? Do you remember? What was the enzyme that got released from the chief cells of the stomach that will be responsible for the digestion of the proteins? Chief cells, enzyme. You remember? Pepsinogen. And this is the inactive form that will get activated by the hydrochloric acid that will get released from the parietal cells. All right? The parietal cells are the ones that will be releasing the intrinsic factor. Remember, intrinsic factor is not a digestive enzyme. It's needed for you to be able to absorb vitamin B12. And cholecystokinin, it's a hormone, not an enzyme, that gets released in the blood. And it enhances the motility of the gallbladder for it to contract, releasing the bile into the duodenum. All right? Gastrin is also a hormone, and those cholecystokinin and the gastrin are going to be released from the enteroendocrine cells. Those are, again, hormones. Again, what is the enzyme that will be helping with the digestion of proteins released from the stomach? It's going to be the pepsinogen. I'm not sure of this information I will be stating. Do you know why Pepsi Cola did get its name Pepsi? Because when they first created it, it contained the digestive enzyme, the pepsin, to help with the digestion. I'm not 100% sure of this information, but I did read this information over the internet, but I'm not 100% sure of whether it's true or not. But many of the websites on the internet say that Pepsi Cola actually was a medication that is created mainly to help with the digestion, and it contained the pepsin, which is the enzyme released from the stomach to digest the protein. All right. All right, so we've got here 3 enzymes released from the pancreas that will be responsible for also the digestion of the proteins. Those are the trypsinogen, chymotrypsinogen, and procarboxypeptidase, and all those enzymes are present in an inactive form. It gets activated when they reach the inside of the intestinal lumen. So they don't get activated in here within the pancreas, they get activated in the small intestinal lumen. Why is it so important to not get them activated in the main pancreatic duct, let's say, or inside the pancreas? Try to imagine you are activating here the digestive enzymes that will be breaking down the meat that you eat. So all the meat that we're eating, it's mainly protein and fibers. So try to imagine that you've got those enzymes activated in here. So what's going to happen to the pancreas if you activated those digestive enzymes inside? It's a self-destruction, exactly. I will be digesting myself. All right, so how come you're not digesting the wall of the small intestine? Which one is thicker? The burger patties that you're eating or the wall of your small intestine? Which one is thicker? The burger patty or the wall of the small intestine? What do you think? What do you think? Which one is thicker? Asking about the thickness on here. Any guess? Yeah, it's the best answer. Yeah, it depends which burger. Exactly. So according to the thickness of the burger, right? All right, but generally speaking, for most of the expensive burgers are gonna be much thicker than the wall of the intestine, right? So how come I'm able to digest this burger, but I'm not able to digest, or I'm not digesting the wall of my small intestine? Because simply, if you remember, the internal lining of all the gastrointestinal tract from the stomach all the way to the terminal part of the large intestine is going to be lined with simple columnar epithelium. And this type of epithelium is going to be producing mucus. It contains mucus secreting cells, goblet cells. So because of the presence of this mucus layer lining the inside of my stomach, lining the inside of my small intestine, those enzymes will not be able to break down the wall of my digestive tract. But you don't have this mucus lining inside your pancreas. So they will only become activated where again? Inside the small intestinal lumen. How do they get activated? The enzymes that will be responsible to activate the trypsinogen are the enteropeptidases that are actually released from the endothelial cells of the duodenum. So there is no way you can activate them outside normally. The only way for me to activate those enzymes is by getting them to on inside the small intestinal lumen. This is where they will get in contact with the enteropeptidases released from the small intestine that will actually gonna be activating them. So the trypsinogen gets activated into trypsin. This is my active form. Trypsin activates more of the trypsinogen. Also, it activates the other two enzymes. It activates the chymotrypsinogen into chymotrypsin. It activates the pro-carboxypeptidase into the carboxypeptidase. So the three enzyme active forms, those are the trypsin, chymotrypsin, and the carboxypeptidase. So how would I be breaking down and absorbing the different types of nutrients? So we're looking on here at a blood vessel. And this is one of my intestinal cells. This is a brush border. Those are the microvilli of my intestinal cell, like this. And the brush border is in contact with the intestinal lumen on the other side. Take it out here. This is my right, so this is my intestinal lumen. What is the different nutrient types that you know? What are the different nutrients that you know that you are actually eating every day? What are the nutrients that you're eating? Do you have an idea? Vitamins, minerals, what else? Proteins, carbs, fats. Yes, exactly. Proteins, carbs, fats. So the first ones that we're looking at now, it's going to be the carbohydrates. Yeah. Iron is going to be part of the minerals. Yes. So looking at first, let's say the proteins. Looking first at the proteins. How those proteins that you're eating going to be broken down? The first enzymes that will be breaking down the protein is the ones that we got released from the stomach, and this is going to be the pepsin. If you remember, it's getting released from the chief cells of the wall of the stomach. Those large proteins will be broken down by the pepsin and the hydrochloric acid into large polypeptide chains. And the large polypeptide chain is going to be broken down into small polypeptide chains by the pancreatic enzymes. And if you remember, the three pancreatic enzymes that will be breaking down the proteins, those are the trypsin, chymotrypsin, and the carboxypeptidase. Those small polypeptide chains gonna be broken down into the absorbable form of the proteins. Those are either the monosaccharide, the amino acids, or the dipeptides. And the enzymes that are responsible to break down the small polypeptides into single amino acids or dipeptides, those are the brush border enzymes, which get released from the intestinal cells. Those are like the aminopeptidases, the carboxypeptidase, and the dipeptidases. So the only two absorbable forms for the proteins, those are either the amino acids, single amino acids, or dipeptides. What I mean by dipeptides, those are two amino acids that are attached to one another. So I can only absorb either single amino acids or dipeptides. For single amino acids, I will be transporting them by co-transport with sodium. So what will I do on here? I will be moving the amino acid from the intestinal lumen to the inside of the intestinal cell along with the sodium. So this requires me to consume ATP to get this amino acid. Same for the dipeptide, but the dipeptides formed of two attached amino acids gonna be transported by co-transport, this time not with sodium, but with hydrogen. And again, this is gonna require me to allow this transport to take place by consuming energy. It's an active membrane transport, co-transport with hydrogen. For the dipeptides, the two amino acids attached to one another gonna be broken down using this hydrogen that got transported with them. So now what I have inside my intestinal cells are just single amino acids. So this creates a high concentration of amino acids within my intestinal cells compared to my blood. I have those channels and carriers that will be allowing the amino acids to travel freely down their concentration gradient. So here I'm allowing the amino acids to travel from the intestinal cell into my blood by diffusion. And if you remember, what is the name of this type of diffusion? It's a facilitated diffusion. Why do I call it facilitated diffusion? Because it required a carrier here to allow the amino acids to travel down their concentration gradient. So did you consume any ATP on the other side of the cell, at the basal membrane of the cell? Did I consume any energy to allow the amino acids to travel from the intestinal cell into my blood? Did it require any energy? What do you think? Did I need any energy to allow the transport of those amino acids from the intestinal cell into my blood? No, I don't need energy here. It required me to consume energy to transport the amino acid and the dipeptides to move from the intestinal lumen, from the lumen of the intestine, into the intestinal cell. But as I'm building up higher concentrations of amino acids inside the intestinal cells, those amino acids will be capable to travel down their concentration gradient from the intestinal cell into my blood through their specific carriers. So it's a facilitated diffusion. So looking here, how do I allow the absorption of amino acids? Again, absorption of the amino acids will be taking place by co-transport with sodium, compared to the dipeptides going to be absorbed by co-transport with hydrogen. This hydrogen that you transported the dipeptides in with will be used to hydrolyze those dipeptides into single amino acids within the intestinal cells. Amino acids will then leave the epithelial cells by facilitated diffusion and will enter in the capillary blood to be transported to the liver through the hepatic portal vein. So I'm passing from the intestinal lumen to the intestinal cell, and then from the intestinal cell to the capillaries, to the veins, to the hepatic circulation, where those nutrients gonna be processed. Second type of nutrients, it's gonna be the carbohydrates. And the carbohydrates gonna be broken down first within the oral cavity. If you remember, you start the chemical digestion within the oral cavity under the effect of the digestive enzymes of the salivary glands. And if you remember, one of the salivary gland enzymes, this was the amylase, which is responsible to break down the starch, the complex carbohydrates. Salivary amylase And the pancreatic amylase is going to be breaking down the complex carbohydrates into disaccharides or oligosaccharides. Oligo means few. So I break down this complex sugar into smaller sugars in the form of a few sugar units or 2 sugar units, disaccharides. Most common types of disaccharides, we've got the lactose, maltose, and sucrose. The lactose is formed of a galactose and a glucose. The maltose is going to be formed by 2 glucose, and the sucrose is going to be formed by a glucose and a fructose. Lactose, it got its name because it's going to be present in the milk. Sucrose is the sugars that we find in the fruits, and maltose is the sugars that we find in the starch, like the bread. Those disaccharides are going to be broken down into their monosaccharide forms by the brush border enzymes released from the intestinal cells. An example of those brush border enzymes includes the lactase, maltase, and sucrase. So the sucrase splits the sucrose into a glucose and the fructose. The maltase breaks down the maltose into 2 glucose. The lactase breaks down the lactose into a galactose and a glucose. The monosaccharides are going to be the absorbable form of those carbohydrates. I can't absorb disaccharides, I can't absorb oligosaccharides, I can't absorb starch, of course. So I need to break down those carbohydrates into monosaccharides. So if we're looking on here at how do you allow the transport of those different monosaccharides, again, we're drawing here, this is my blood vessel. And this is the basal layer of my epithelial cell, and this is its brush border. And on the other side of the intestinal cell, we've got here, this is the intestinal lumen. So I've got here the absorbable form of the carbohydrates. I've got the monosaccharides, glucose, galactose, and fructose. The galactose and the glucose gonna be transported the same way like what did transport the amino acids, the single amino acids, by co-transport with sodium. Compared to the fructose, fructose is going to be transported through its own carrier from its high concentration gradient within the intestinal lumen to the inside of the intestinal cell where I have a very low concentration gradient of it. So what do we call this transport? It's a facilitated diffusion. So I've got now the fructose, the glucose, and the galactose inside my intestinal cell. This will create a high concentration gradient of those monosaccharides inside your intestinal cell compared to their concentration gradient within your blood. So they will travel through their specific carriers down their concentration gradient by, again, facilitated diffusion. So which one so far I didn't consume any energy to allow its absorption? Is there any nutrient here that I didn't consume any energy to allow its absorption that you can see? Did you consume energy to allow amino acids, dipeptides to travel? Yes or no? Did I consume energy to allow amino acids and dipeptides? Yes. Did I consume energy to allow the glucose and galactose to travel? I co-transport with sodium. Yes, I consumed energy. Did I consume any energy here for the fructose? No. It passed from the intestine lumen to the intestinal cell by facilitated diffusion. It's a passive transport. And again, it passed by facilitated diffusion from the intestinal cell into the blood. So no energy has been consumed to allow the absorption of the fructose. So let's read this part on here. The glucose and galactose are going to be absorbed by co-transport with sodium ions. The fructose is going to be passing by facilitated diffusion. All the monosaccharides now are kept inside the epithelial cells, will pass by facilitated diffusion to enter the capillary blood and to be transported again to my liver through the hepatic portal vein. Any questions? Any questions? All right, so moving on to our third type of nutrients that we're consuming. Those are the fats. Fats needs to be emulsified first before they get digested. And the emulsification of the fat, it's gonna be taking place by the bile salts contained within the bile. This emulsification will allow the fat to become more soluble, more water soluble, getting more exposure to the digestive enzymes. The main digestive enzymes that would be breaking down the fats are going to be the pancreatic lipases, and they will break down the fat into two main structures. Those are the fatty acids and the glycerol, or monoglycerides and fatty acids. How would I be able to allow the absorption of those fats? Again, we're looking here at the intestinal lumen. This is my intestinal cell on here, and this is going to be the brush border of the intestinal cell. This time I'm not drawing a blood vessel, instead I'm drawing a lymphatic vessel. So this is not blood, this is lymph. So I've got here glycerol and fatty acids. They will be able to travel by simple diffusion. They don't need any carrier. They don't need any energy or any channel to travel into the cell. Why? Because simply, if you remember, the plasma membrane is formed of phospholipid bilayer, and the main content here is going to be fats, and it's going to be fat soluble. So the fat will be passing by simple diffusion. I don't need channels, I don't need carriers, I don't need sodium to allow my co-transport. I will simply diffuse through the plasma membrane. We call this transport is a simple diffusion. So glycerol fatty acids, they are lipid, so they will pass through the plasma membrane, which is able to allow any lipid soluble substance to travel. The problem now is that this glycerol and fatty acids are not water soluble, so they can't simply leave the cell and travel in the even lymph or blood. They are not water soluble. They will start to precipitate, they will accumulate, and they will block the circulation. So what will I need to do? I will need to make them water soluble. So how can I do this? I will first try to neutralize those fats. I will form neutral forms of those fats. And the neutral form of the fat on here is going to be my triglycerides, in which I've got a glycerol attached to 3 fatty acids. I form the triglycerides, but it's still fat. It's not water soluble. Yes, it's a neutral fat, but it's still not water soluble. So I decided to add protein to the triglycerides, and this protein will make it more water soluble. And I add other triglycerides to this protein. Triglycerides attached to proteins which contain charges, and those charges are what's gonna allow the triglycerides to become more water soluble. But now I've got a huge chemical complex. I've got triglycerides attached to proteins. The triglyceride part is water insoluble. The protein part is water soluble. For them to be able to pass and circulate into my lymphatic vessels or blood vessels. So now it's becoming a huge chemical compound, and there is no way that all this structure gonna be able to pass through a carrier or a transporter available in the plasma membrane. So the only mean for me to allow this structure here, and we call those structures are my chylomicrons. Chylomicrons. The only way for me to allow those chylomicrons to be released to the outside, I will store them in vesicles, and I will make this vesicle to fuse with the cell membrane, releasing the chylomicrons to the outside. Again, what is the structure of the chylomicrons? It contains triglycerides attached to proteins. And then from the lymphatic vessels, those triglycerides attached to proteins will find their way to be conducted to the blood and then to the liver where I will be able to modify and store whatever I. So again, how do I absorb the fats? We've got the absorbable form of the fat. It's going to be the fatty acids and the monoglycerides. Fatty acids and monoglycerides are going to be passing by simple diffusions. They will pass from the intestinal lumen to the intestinal cell by simple diffusion. Nothing to worry about in here, as they have a higher concentration gradient within the intestinal lumen. Fatty acids and monoglycerides are going to be combined to form a neutral form of the fat. This is called the triglyceride. Those triglycerides are going to be attached to other lipids and proteins within the cell, and we call the whole structures that gets formed, it's the chylomicrons. Chylomicrons are going to be released from the intestinal cell into the lymphatic vessel within the intestine by exocytosis. And if you remember, exocytosis is an active membrane transport. It requires ATP. Chylomicrons will enter the lymphatic vessels in the villi of the small intestine and will circulate through the lymph in the thoracic duct. Some short chain fatty acids are absorbed and move into the capillary blood in the villi, and those going to be transported directly to the liver rather than going into the lymphatic vessels. So only the short chain fatty acids are the ones that will go directly to the liver. Any questions? Questions? All right, so moving on to our last type of nutrients today. This is going to be the nucleic acid. What is a nucleic acid? If you remember, nucleic acids are going to be present in the DNA and RNA. If you remember the structure of the DNA and RNA, it was formed by a pentose sugar, a 5-carbon sugar, either in the DNA deoxyribose or in the RNA a ribose sugar. Attached to it, it's a base. If you remember, we've got the purines and pyrimidines, adenine, guanine, thymine, cytosine, or uracil, and a phosphate group attached to the ribose sugar or the deoxyribose sugar. So this is the building block that we've called the nucleotide of the DNA and RNA. So actually, those are also the absorbable forms of the nucleic acid. So you absorb those phosphate ions, you absorb the pentose sugar, you absorb those nitrogen containing bases, adenine, guanine. So you split them apart and you absorb each one on its own. The enzymes that will allow you to break down the nucleic acid, DNA and RNA, this includes the ribonuclease and the deoxyribonuclease. Both are released in the pancreatic juice. We've got some other enzymes that will be breaking down the DNA and RNA and released from the intestinal cells. Those are the nucleosidases and the phosphatases. Units of the nucleic acids, ribose, deoxyribose, bases, phosphate will enter the intestinal cell by active membrane transport through the carriers, and then will be transported from the intestinal cells to the blood, and then from the blood into the liver through the hepatic portal vein. So there is no cheap way to move those nutrients from the intestinal lumen to the circulation. So it's all going to be conducted by active membrane transport. Questions? Questions? All right, we can have a break for 10. We come back, discuss the large intestine. Any questions before we go for a break? All right, we'll see you in 10.